Welcome to The Explainer. Today, we're literally opening the hood on a total masterpiece of biological engineering, carbohydrate metabolism. We're going to go straight to the core of how the human body processes fuel at the cellular level by examining the powerhouse pathway known as glycolysis.
Think about it. Every time we eat, our metabolism is hard at work transforming dietary fats and carbohydrates through a highly regulated, totally interconnected series of chemical reactions. And the ultimate end goal of this whole process? Converting that raw material into the body's primary cellular currency, ATP. ATP operates as the central distributor of energy for literally every cellular function, while metabolic byproducts like carbon dioxide and water are efficiently cleared out. It is a continuous, perfectly tuned cycle.
Okay, let's dive into this. Here's our clear, step-by-step flight plan into the microscopic world of cellular biochemistry today. 1. Meet your metabolic engine. 2. Glycolysis, the universal pathway. 3. Investment and payoff phases. 4. Aerobic versus anaerobic conditions. 5. Calculating the energy gain. And finally, number 6, what breaks the engine?
All right, Section 1. Meet your metabolic engine.
So, this incredible engine of ours really balances on two fundamental, highly regulated states of building up and breaking down molecules. On one side, we have anabolism, which is the synthesis of biologically important compounds. Now, this is an endergonic process, meaning it requires a direct input of energy to build those complex structures. On the flip side is catabolism, the oxidative breakdown of complex molecules. That's an exergonic process, so it releases energy. Essentially, catabolism burns the fuel to extract energy, and anabolism utilizes that exact energy to construct and repair the structural tissues our bodies need to function. It's an elegant balance, right?
Moving on to Section 2, glycolysis, the universal pathway. The name glycolysis derives from the Greek words glykos, meaning sweet or sugar, and lysis, meaning to split or break apart. So it translates literally to the breakdown of sugar. And what makes this primary catabolic pathway so utterly fascinating is its location. All of the glycolytic enzymes driving this process live right directly in the cytosol of our eukaryotic cells. It doesn't even need to be tucked away inside a specialized organelle like the mitochondria to get started. The whole process fires up right out in the main cellular fluid.
Now, what's really interesting about this abstract biological concept is that breaking down glucose isn't just a single blunt strike of a hammer. Actually, it's a perfectly choreographed sequence of exactly 10 enzymatic reactions. It takes 10 highly specific, interconnected steps to properly extract the energy required to power our biological systems.
We start with a single 6-carbon molecule of glucose, which transforms entirely through this precise sequence into 2 3-carbon molecules of pyruvate. But the true value, the real magic, lies in what happens along the way. The process converts ADP into our cellular currency, ATP, while also reducing NAD+ into NADH, which carries vital electrons for later use. It's just a masterpiece of biological transformation.
Let's move to Section 3, investment and payoff phases.
Think of these 10 steps kind of like a financial transaction. In biology, you often have to spend cellular currency before you can reap the biological profits. So phase 1 is the investment phase. Here, the body actually utilizes its own ATP to phosphorylate glucose, eventually converting it into fructose 1,6-bisphosphate. You're spending cellular currency right out of the gate to prep the molecule. Then comes phase 2, the payoff phase. This is where glyceraldehyde 3-phosphate is ultimately converted down to pyruvate. And in doing so, ATP is finally released back into the system. The biological profits are officially reaped.
Within that payoff phase, there's a moment of absolute finality. The crucial step is the irreversible reaction where a molecule called PEP forms pyruvate, and this is catalyzed by the pyruvate kinase enzyme. This step operates as a strict one-way street. Once you cross this line, ADP is phosphorylated into ATP, officially cementing our profit. This biological finality is incredibly important because it prevents the body from wasting valuable resources trying to run the cycle backward. It's literally a masterclass in metabolic regulation.
Section 4, aerobic versus anaerobic conditions.
And this brilliantly illustrates how dynamic our metabolic engine is based on the local environment. When oxygen is plentiful, under aerobic conditions, the end product of glycolysis is pyruvate, which can then go on to generate massive amounts of energy in the mitochondria. But consider a scenario where you're sprinting, right? Your muscles run out of local oxygen. In an anaerobic state, the engine doesn't just stall out, no way. It dynamically shifts gears, altering its final step to produce lactate instead. This adaptability keeps the core engine running and ATP flowing even when the optimal fuel conditions suddenly change.
Up next, Section 5, Calculating the Energy Gain.
So, the crucial point is, what is the ultimate biological bottom line of this entire metabolic investment? It's exactly 7. When one molecule of glucose is fully oxidized to pyruvate under aerobic conditions, the net energy gain for your body is exactly 7 ATP. This accounts for the ATP directly gained, minus the ATP we invested earlier, plus the ATP equivalent generated from those NADH carriers produced along the way. That 7 ATP is the hard-earned profit your cells use to keep everything functioning.
Which brings us to our final stop, Section 6. What breaks the engine?
Let's add a touch of toxicology intrigue here, because any complex engine is vulnerable to sabotage. Specific chemical inhibitors can easily hijack this delicate 10-step sequence. Take arsenate, for example. It acts as a biological imposter. Because its structure is incredibly similar to phosphate, it sneaks into the pathway, taking phosphate's place but yielding absolutely no energy. The result? A net ATP profit of 0. The cell does all the work for 0 payoff. Similarly, sodium fluoride specifically targets and blocks the enolase enzyme, completely disrupting the pathway. And finally, iodoacetate acts as a fatal enzyme inhibitor that totally jams the gears, halting glycolysis entirely. It just highlights how precise and delicate this entire sequence truly is.
We've seen how external chemicals can break the engine. But what if the blueprint was flawed from the very start? Consider the biological consequences if someone were born with a genetic deficiency in the hexokinase enzyme at the very beginning of the pathway, or the pyruvate kinase enzyme at that crucial, irreversible final step. Think about it. How might missing just one specific microscopic enzyme out of 10 completely rewrite an entire body's energy story? Pretty wild to think about, right?